I think we'll go ahead and start, and I'll just kind of introduce Dr. Judy Woo Smart here. We're lucky to have her join us from the University of Nebraska's Lincoln Bee Lab. I've been a big fan of her work ever since she came down to K-State back when I was in graduate school and gave us a lecture. And so today she is going to deliver the second lecture of our three-part webinar series on neonics. And if you missed the first lecture from Dr. Mike Bredesen, you can find it on Ecdysis's YouTube channel. But one of the topics that Dr. Bredesen discussed was the movement of neonics via water through the ecosystem. And Dr. Wu Smart is going to kind of pick up that torch where Dr. Bredesen left off last month. She's going to dig into the subject much deeper today, discussing the unintended consequences of this pervasive pesticides on environmental, animal, and human health and what we can do to help. So with that, I'll turn it over to you, Dr. Wu Smart. And yes, please ask questions as we go, and there will be opportunities to have Dr. Wu Smart answer those. So put them in the Q&As.
Thank you. What I'd like to do is, as I'm pulling up these slides, I'm an apiculturalist. I study bees. I study honeybees predominantly, but I also work with wild bees, bumblebees, and others. And this talk is kind of a combination of multiple studies, kind of pulling together this body of knowledge related to neonics and pesticides in general and then kind of pulling in the connections to the broader ecosystem. And so some of you might not have heard of this term One Health, but really to get started, this is one of our apiaries with cows just running through the apiary, but you can see that we are living in landscapes where there's a lot of interaction amongst the wildlife, the livestock, the humans, and the environment. And so there's a lot of things that we have to consider in terms of the things that impact our system. And so sometimes we talk about these as grand challenges or key global issues related to food security, loss of biodiversity, changes in our environment. But in One Health, we basically lump them into three buckets. And the understanding is that the health of humans, wildlife, and the environment are all interconnected. And so when we are addressing these issues, we really do have to look at them through that One Health lens to look at all the connections. Because it's not just the loss of pollinators and other beneficial insects, which I'm going to focus on, but that loss affects humans, other wildlife, and is an indicator of the environmental quality. So that's kind of what I'm going to try to pull together with what happened in Nebraska. Ryan alluded to the pesticide issue, and then kind of expanding beyond that, what we've learned over the last few years and pulling all the knowledge back together to kind of look at it as a whole system. And so fundamentally, we have this challenge of wanting to protect the good bugs, but wanting to also remove or control the bad ones. And so that's where there's a lot of conflict between how do we protect pollinators, bees, and natural enemies, while also dealing with the insects that eat our crops, that invade our homes, that spread diseases. And so there's a lot of issues related to just trying to figure out the balance between those. And while I'm going to pretty much focus on honeybees, I do want people to recognize that there are, probably about 4,000 types of bees in North America, 20,000 worldwide. We think there's probably about 4 to 600 different species in Nebraska. And there's a lot of diversity amongst the bees. You'll see bumblebees, there's a number 49 there, and honeybees is the number one. And that just represents that there's one species of honeybees that was imported into the US. There's 49 species of bumblebees. And so you can see the number of species amongst these other groups, 400 plus in minding bees alone, that really kind of is often unknown to the general public or to folks that love bees but only really think about them in terms of bumblebees and honeybees. So there's a lot of diversity, a lot of bees that utilize soil or plant materials for nesting and habitat. But this talk will focus mostly on our managed livestock, our honeybees. In general, though, all of these bees are going out in the environment collecting nectar and pollen for their own development and growth and energy reserves. And that interaction with plants causes pollination to occur, and that's why they are often very useful, effective pollinators for our own cropping systems. But they also pollinate a number of native and wild plants that are important for the environment. Generally, these types of facts have been kind of widespread and well known that 1/3 of our diet is pollinated by insects. But a lot of people don't realize that 80% of that is done by managed honeybees moved by their beekeepers. So there's this a big industry that, or a big support structure behind our food cropping systems that a lot of people don't realize that is really, you know, supported by the bees and the pollinators that are servicing these crops. That includes a number of the fruits, the nuts, oilseed crops, vegetables, and even livestock feed and like alfalfa and vetch. So there's a number of things that the bees contribute to. The pollination by these insects actually increases the yield, uniformity, and size of some of these foods. And generally, it is the most nutritious foods that we rely on. So, in general, you can see the image of a strawberry plant that requires cross-pollination by insects. If it doesn't have that, those fruit become aborted or abnormally shaped and don't grow right. And so, we generally give people that message that if we want to live healthy lives ourselves, we have to maintain healthy bee communities. And this is another kind of a view of some of the things that often people don't realize is that the beekeepers that are managing these bees, commercial migratory beekeepers, are moving these hives to meet the pollination demands of a lot of different crops. California almonds is a major industry that supports the beekeepers, and a lot of the colonies go into almonds for pollination in that January, February, March time point, and then they move into different cropping systems. And then during the summer, they also require summer feeding grounds, which is forage, canola, alfalfa, sweet clover, sunflowers. These are really critical flowers that bees rely on when they are not meeting the demands of these pollination contracts. So when we're talking about pesticides and pesticide risk, I wonder if I can hide this video panel. There we go. Now it's not in the way. Um, estimating field relevant exposures is really difficult. We talk about how and where the bees are getting exposed, and that could really depend on whether you're talking about an urban landscape or an agricultural landscape. There really are different types of risks and different chemicals being used in these, but we can't say that one setting is higher risk than another. It really is location dependent and who's using what chemicals to control what. It is kind of different in when we're studying ecotoxicology and with bees in particular. It's difficult because we need to understand not only where they're getting exposed, but what they're bringing back to the hive. Because these are social insects, there's a lot of interactions and social behaviors that are critical for their own, here it is. Here, I got rid of it. For their own use and control, the social communications and the roles that they play in the hive are highly evolved and specialized. And so, for example, honey or nectar being brought back from the hive might be distributed and fed throughout the colony so that all nest mates and the young are fed honey. But there's only a small portion of bees that are actually bringing back pollen and feeding on that pollen for brood. And so depending on what is being brought back to the hive, is going to change who is being exposed to those contaminants. And so it gets very complicated very quickly. Um, Essentially, the last, I don't know, 12, 13 years of looking into how pesticides affect bees, we can generally say that there's these acute lethal exposures that might cause individual death of a young bee, which is that little caterpillar-shaped sea larva there, or an adult bee. It can also have colony level effects where the entire colony is affected, dies, or is unable to function properly. And then when we talk about exposures and effects, we often refer to them as direct effects or indirect effects. And a direct effect might be the compound interacts with the bee and it dies. An indirect effect might be if the compound changes behavior, development, reproduction, or alters the habitat in a way that makes the bees less successful, but isn't directly affecting that individual. So there's a number of different things that could happen. However, the regulatory focus often focuses on whether or not it kills a bee or a colony. And so it kind of, while there's a lot of studies that show these chronic sublethal exposure impacts, less of that, those studies are emphasized in regulatory actions and changes. So when we talk about neonics, I'm going to bring up a lot of the American Bird Conservancy resources and studies. They are a group that has been actively working on these neonic issues and concerns as long as the bee groups have as well. So there's a number of great resources that and work coming from the American Bee Conservancy. But neonicotinoid pesticides are, as the previous speaker mentioned, are synthetic nicotine compounds. And I'm just going to briefly go through what they do and how they work just so that people are all on the same page. But they basically target and bind to nerve cells. We have these nicotinic acetylcholine receptors that can receive those. They bind to it and it causes constant transmission of these nerve signals. That can lead to cell exhaustion, paralysis, and death. But with these compounds, they have a higher affinity or selectivity for insects. So they're more toxic to insects while they're considered safer for mammals, including our human applicators. So that's oftentimes why these class of insecticides are favored within the pest control group, but folks. But one of our main concerns is that they do act systemically, meaning that they can move to all parts of a plant, including the nectar and the pollen is where we are most concerned with bees foraging and getting into contact with them. Now, there's a number of active ingredients within this class of pesticide insecticides, and then hundreds of trade names. So these are the formulated products that are sold in the shelves of, available for farmers and homeowners to use. But you really have to kind of dig into the active ingredient to really know what type of neonic it is. The trade names don't always give you that information up front, but the ingredients, the active ingredient list would. Now, these neonicotinoids can be applied via seed, via foliar spray, tree injection, right into the irrigation system. So they're quite versatile in terms of in terms of their application and use. I'm going to mostly focus on these treated seeds, which generally includes a neonicotinoid insecticide as well as one or multiple fungicides. And that is coated on the outside of the seed before so that they're ready to be planted with the chemical kind of permeating that small plant and the seedling that comes from that seed. So that's basically this is kind of a diagram of what we're talking about here. It's applied different ways. If it gets into the seed treatment, it can be expressed in the leaves and then the reproductive structures of that plant where the pollinators are foraging on. And where we're concerned is that this class of chemicals tends to be more water soluble, and there's a lot less known about their environmental fate and spread. It can be through the dust particles that are coming off of the seed or build up in the soil or run off in the water source. And so there's a number of ways that it can easily move off of a target field or off of a target plant and into places where it's not supposed to. This study is from Minnesota when I was a grad student, but this is an example of a small colony that has the ability to fly in and out. This is just an observation hive. And I'm going to pan over to where the colony is being fed 50 parts per billion imidacloprid. It's a pretty small dose, and it's being fed 50 mils or 1.6 fluid ounces every other day. And what you can quickly see is that the bees here are not active. They're alive, but they're not going in and out of the hive foraging, not caring for brood, very little activity, almost like they're asleep. Whereas I'm panning over here, this is the control colony where you see a lot of activity, a lot of bees functioning. And so, what happens if a colony cannot forage correctly, care for a brood correctly, or lay eggs? It can slowly dwindle and certainly not have enough food source to take them through the winter. So this is a great example of how a pesticide doesn't have to outright kill a bee to harm the colony, to make it more less likely to survive a winter. These videos here is just another example of exactly what happens to the queens. If they are being exposed on the left-hand side, you see a blue marked queen actively walking around the hive, and she's supposed to be laying about 1,000 eggs a day. And so she has to actively search for an empty cell before she deposits it. On the upper left-hand side, you can see the abdomen or the hind end of a queen who has apparently stuck her head in a cell and fallen asleep or become inactive. And so this is one of those really stark behavioral things that we have seen neonicotinoids do to these reproductive queens in both honeybees and even in bumblebees, is that it inhibits their ability to move around, and to lay eggs efficiently. And so the image down on the lower right or lower left shows where a queen that is not dosed with neonics would travel quite far, exploring that whole frame and looking for multiple places to lay, whereas a queen that is dosed with a small level of chemicals, and she's not even directly dosed, it's being fed to the colony and slowly being moved into or transferred to her or fed through nestmates. And you can see that many of them just kind of spin around or don't move very much at all. So again, pretty dramatic effects on a reproductive individual. And when we look at things like egg laying and brood production, with honeybees, it's very they have a very strict developmental time. It's a really great model system to study the effects of pesticides because there's so much known about their life history already. They're much like butterflies, they go through an egg, a caterpillar, and a cocoon stage. And in this image, those three stages are shown at the top. And what we see is that the queens are inhibited in laying, but it isn't just the queens and their egg laying, but the brood the offspring that ultimately result becomes spotty and very ununiform. And that brood pattern is an indication of the health of the brood as well as brood care within the colony. So as the doses of neonicotinoids increases, you can see where 0 versus 10 parts per billion, 20, 50, and 100, there's a pretty dramatic increase of the spottiness and the reduction of brood in those hives. Um, Another really kind of alarming thing is that with honeybees, they struggle a lot with a varroa mite, a ectoparasite that feeds on the developing bees at their cocoon stage. And in this diagram, honeybees are uniquely adapted to this disease in the sense that they can sense a mite-infested bee and can remove it before it becomes infectious or comes out and starts propagating new mites. But in a neonicotinoid dose colony, that hygienic removal or that hygienic behavior performance is significantly reduced when they are exposed to these higher levels of chemicals, making it more difficult for beekeepers to control the mite loads in their hives. And that is one of the major problems that beekeepers face within their own hive management. So going back to this diagram, a lot of the times when we refer to a pesticide or a bee kill, it's referring to that acute lethal exposure. But nowadays, with these new chemicals like neonics, we're having discussions and concerns related to pesticide exposure incidences, where it doesn't outright kill the bees, but it's altering their behavior, affecting their development, their reproduction, and that altering the habitat that makes them less successful to survive as a unit, and particularly over the winter. And we're also seeing that the ramifications of this is that the bees are dying off in the summer as opposed to having most of the losses over winter. You know, we're seeing more and more of these colonies dying when they shouldn't, when they should be foraging and being very productive. So when I started at University of Nebraska Lincoln in 2015, I set up these hives in all the wonderful honey producing locations that Dr. Marion Ellis, my predecessor, had pointed out. And unfortunately, I couldn't keep bees in this one location alive. They kept spilling out in massive, like a classic acute toxic kill or acute lethal exposure. And I would even vary the time of placement of moving hives in to see if it was related to crop planting exposure and the dust off that might come from the field being laden with some of these chemicals. And that really didn't seem to be what was affecting the colonies. You can see that even within 24 hours of collection, quite a lot of those bees were starting to pile up. Inside the colony, you saw a lot of loss of bees at the immature stage. I even found adult bees with their heads in the nectar cell dying as they were either eating or processing that food. And so it was clear at that point that it had to be associated with the food source, not necessarily the mite and diseases that are common amongst honeybee colonies. And my student at the time in this location was also setting up an experiment to look at the ramifications of this dust plume. So when pesticide-treated seeds are being planted in the field, if it is windy or the conditions are right, that field dust blows across the landscape. And so the concern was that a lot of the habitat that we were starting to establish for pollinators and beneficial insects was then going to be blanketed by these chemicals. So the study was looking at the role of drift barriers and whether or not those could be natural mitigators of that dust drifting into those habitats. So strategically placing habitat near those drift barriers might reduce those loads. So her study um, um, established these habitats all around our research stations, and then she did a number of things to look at the chemicals in the habitats as well as try to quantify that dust plume. And so that's kind of how we started to figure out that there were some really interesting things with the bee losses and the preliminary pesticide results. And in this example, this is a tree line. We were finding that the milkweeds that we were collecting closer to the waterways was higher than the ones closer to the fields, counter to our hypothesis. And these milkweed plants were showing levels as high as 3 to 5,000 parts per billion, which honestly we contested for a couple years. We resampled, we sent it to a different lab, and it turns out that they were that high. This is really unheard of in terms of the literature. Looking at the various different residue studies, um, uh, neonicotinoids can vary depending on the plant and how it was applied. So generally, treated seed applied crops only yield just, very low levels of residues in the pollen and the nectar. If it's soil applied and it's an herbaceous plant, those levels get a little bit higher, but they're certainly much higher when you are talking about those more woody species, shrubs and trees. And so Cornelian cherry has been shown to harbor residues as high as 3,000 parts per billion when it was soil applied. And even if it's water applied within the same plant, it can vary whether or not you're collecting pollen versus nectar. So it does complicate things, and when we're trying to understand exposure, when the plants themselves respond to these chemicals very differently. So with milkweed leaves as high as 5,000 parts per billion, we knew that those were going to be levels that are lethal to insects that would be feeding on either the leaves or the nectar or the pollen. So this is when we started to reach out to the state and federal officials. We connected with the Department of Ag and actually the Department of Energy and Environment, as well as EPA officials, to talk about what was going on with these dying bees and ask. And honestly, I was asking for help to assist with the testing and the investigation. To my surprise, because I spent a year or two ruling out the on-farm applications, they were unable to assist in those bee investigations. And the reason why is because these pollinator protection policies and resources fall under FIFRA, which is the Fungicide Insecticide Rodicide Act, right? It's a federal act that oversees the sales, use, application, and disposal of pesticides. It doesn't really handle pesticide pollution. And that's where this case kind of falls in the gray area because we felt that this was more of a pollution problem, probably associated with the waterways, not some misapplication of the chemicals. And they really gave us feedback saying that, it'd be better to have more hives on site because we just had a limited number of colonies. And we were tasked to kind of figure out where the bees were getting exposed to which compounds, what levels, and where. And that really requires intensive sampling from the bees to what they're collecting to their surroundings. And so that kind of is what we've done. After 3 days after our meeting, I put out 12 additional strong colonies, set them out various locations, 1 to 3 miles away, and then proceeded to document their loss. They really told us that at this point, they let us know there was an ethanol plant roughly 2 miles north of our honey house, which, our apiaries are marked here in pink dots. And those waterways, continuous and intermittent ditches were what we were really kind of focusing on, knowing that the milkweeds closest to the waterways had the highest levels of chemicals. And even the milkweeds along these waterways, when we were looking and sourcing these milkweeds, we were finding dead bees that looked like they were collecting glutation or that water from the milkweeds dying mid-foraging. And those colonies that we set out, within a week, we began observing these high losses again. And so you can see on the left-hand side, the extent of those losses versus in all of our control colonies around multiple cities, we did not see that extensive loss of bees. Now, the ethanol plant that they alluded to that said had a number of violations, they said were using pesticide-treated seeds as a primary source of carbohydrate. And I know nothing about ethanol. So we had to go through and learn how that process even occurs and what was happening. And normally, grains are received from these plants, go through a milling, cooking, and fermentation process. And that creates both liquid and solid byproduct waste that is typically fed to livestock. They're known as distiller's grain as a feed supplement. But given that these are contaminated with pesticides, this company was offering those, both the liquid and the solid, as soil conditioners for farmers to place in their land as an amendment to their soil health, without disclosing the chemical residues. And they argued that they were not responsible for containment or disposal of these hazardous waste products because they didn't create waste, but rather they created economically viable byproducts that farmers could use, i.e. the soil conditioners. So there's a lot of interesting things that were going on in terms of the regulatory policy side of things and what was legal, what isn't legal, and some of the loopholes that allowed this whole thing to happen. So in the ethanol plant, this alt-ent facility, There was a number of surplus treated seeds that are no longer viable for the market. So again, these are not from the farmers. These are from the seed companies being used as their primary energy or carbohydrate source for production of ethanol. And so just to give you a sense of the sheer volume of this, many crop seeds are pre-treated with a mix of these fungicides and neonicotinoids. The farmers often buy treated seed by default and not as a customized decision per field, like whether it needs it or has these soil pests that could be problematic. So treated corn seed, if it's older than 2 years, generally are not sold because of concerns with reduced germination. And so in 2019 and 2020, there was flooding, massive flooding in the Midwest both years, kind of preventing some of the corn to be planted early because the fields were too wet. Um, so that leads to excess seed. But in this plant, other crop seeds and harvested grains were all being processed and mixed in as well. So there was a number of things going in through this production plant. On the downside is those byproducts were then being dumped outside without any protective measure to prevent from runoff or leaching of those residues. So as I was following up those waterways, it led me to a number of these huge waste piles. Some of them were 7 to 9 feet tall. They exceeded my height, and you could see puddling from anytime it rained. This is a picture I took after a major rainfall to just show how all of that waste gets run off and then right into the culverts and then moves down off that property and into our research property. And so this is a great video created by Nebraska Stand Collective that kind of just shows a drone imagery. It's a nightmare. Can you hear this? So this is a great time. If you've got questions, you can put them in the chat. I'll try to address it. It's just like a 4-minute video here. But it shows kind of this plan, as there was a lot of inspections and violations and kind of scrutiny. A digester broke during the winter and kind of spread all this fluid down these ditches. And beyond those trees is basically where our research facility is. So they interviewed one of the residents that had been really concerned.
They thought with field corn. I'm not sure when they changed to seed corn. The town was never aware of any permission to do this. We have our own well for water, and they hit water at 40 feet. And in a million years, you're not going to convince me that after 2 or 3 years of all of this toxin leaching into the ground with no barrier, that groundwater won't be contaminated to a degree.
So what's floating in that lagoon are damaged liners.
Them installing one of the liners in the lagoons when they first did the lagoons, and the liner was torn when they put it in. It was already torn. So it's been leaking forever.
Up to 98%.
Yeah.
All treated seed corn in North America, not just the United States, but like Mexico, the United States, and Canada was being funneled into Little Mead, Nebraska, a village of like 700 folks, 500 to 700 folks, right?
Yes.
Poisoning. I mean,
And you can see cattle being right adjacent to that property.
Large manufacturers like Bayer, Syngenta, multi-billion-dollar agricultural facilities.
Yep. Well, ground conditioner is not regulated. It's not regulated like fertilizers, etc. are. So when they labeled it ground conditioner, they were able to bypass having to have all these regulations put on them, and no department knew exactly what to do.
This all began sort of with the bees. Finding the collapsed colonies and the manner in which they were finding the dead bees, they were having their tongue stuck out. That's kind of a neurological type of sign that the bees were experiencing.
I have coughed for 3 years, coughed and congested for 3 years. I finally went to a specialist. He said he was pretty sure it was environmental.
Yeah, so I'm going to stop it there. You're welcome to look at it some more, but basically, a lot of the folks that were in this area felt that this, oh, I can't get this off. How do I get this off? There we go. A lot of the folks that felt that this was a health issue were told that these were allergens and not something that they had no idea that pesticides would be something that they would have to look into. So it was unknown to them what was causing these issues. Oops, sorry, I'm trying to get rid of my floating panel. There we go. So in that lagoon that you saw, it was estimated that they were getting rid of 100,000 gallons of pesticide-laden effluent or liquid waste per day, or producing that much. And the concentrations that we're seeing were as high as 58,000 parts per billion in the solid wet cakes, which they were piling all over the property. Some of it was 100,000 parts per billion. Another report from the soil conditioner, I'm trying to get rid of that video plate. The soil conditioner showed 554,000 parts per billion. So it's not just the neonic, but there's also several fungicides and herbicides present. And the NDA estimates that about 33,000 tons of the wet cake was distributed to local farms without their knowledge of those chemicals. So when they issued the stop use, they estimated that what they were recommending as an application would be 85 times higher than their maximum annual field load per year if it was a typical registered product label. So that just shows how hot of a compound that was. And the mixtures of these chemicals are really unknown. And so that's the part where it gets kind of scary is we know that there are these synergistic and additive effects, but we don't know what combinations and to what extent those effects may cause issues. Now, in some of the older documents, it was suggested that birds swallowing an entire seed might get something as high as 100,000 parts per billion. This process was actually concentrating those worse, those levels to 5 times greater than than some of the worst case scenarios that they were projecting. When we went back and actually sampled the soil from some of these private farms, even 2 years after it's been in the environment, we were still finding levels. Of course, they were lower in the soil column, but we were still finding levels, anywhere between over 10,000 parts per billion, and a number of fungicides as well. In the wildflowers, in the pollen and the honey that we were collecting, luckily not a lot in the honey because these are being really harbored and sequestered in the pollen and the comb, but wildflowers certainly had a lot of contaminants in them. Our sampling of the combs also showed pretty high levels in the pollen, higher than what we would expect to be what we would normally find in a colony when we're sampling colonies outside of this general area. Now, I'm having some weird lighting issues here, but okay. So the Serbi, the student I had looking at this dust plume issue and air contaminant, she had these sticky traps to collect airborne particles. And when we asked her to reevaluate the exposures based on those, we found that some of those levels were as high as 500 parts per billion, about a mile away from the plant itself. Certainly higher than the lethal levels, and even at levels that would be concerning to humans and other wildlife. And so when you look broadly at this plant itself, we were finding contaminants in the air, in the soil, in the plants, and the water. They were still soliciting for more seed. They're calling it a green recycling program, despite multiple notices and violations. And of course, they were self-touting that nearly 98% of all discarded seed from North America was being funneled and processed through this plant free of charge. Just bring it in, pay for your own transport. So on our end, we were finding these sick bees, but we're also seeing evidence of other wildlife feeding on the dead bees. So on the left, you see what we refer to as meat wads, and these are just skunks that will eat up the dead bees, suck out the juicy parts, and then spit out the cuticles. And so that's what you see is these clumps of dead bees being sucked out, chewed up, and then spit back out. On the right-hand side, I think it's if I remember correctly, it's a juvenile horned lark that is acting very peculiar. Generally, they are not so comfortable near people. So I don't know if it was cognitively impaired, starving, or what was going on. But there's actually two humans standing right on that's my foot. And on the other side of the hive is my technician that is also there. So this bird seems to be completely unaware of our presence, or just acting very peculiar in a sense. Now, like I mentioned, the video showed the digester busting and releasing a lot of this waste into the streams. We took some videos of migratory birds just waiting in it, and they've been in there actively. They're migrating through, they see a pile of water, and they go in, they start feeding, and they start cleaning themselves. This is very difficult to kind of actually understand what the impacts would be on these migratory birds, because as soon as you approach them, they fly off, and there you have it. You don't know how long they've been in there, what they've been eating, and how long that exposure is. But what we do know is that the residents, like Jody, who was in the video, they were complaining for years and were told that it was likely an allergen or some kind of response to something, a response to an allergen, whether it's from the plant dust or some other fungi, mold, or something else that was growing on the property. But they really didn't think about the chemicals or know about the chemicals. So when we started to kind of make people aware of the losses of bees, that was clearly an indication that it wasn't allergen related, but there's something deeper. And we started connecting with a lot of number of people. This allowed us to get a lot better sense of the facility and the environment as you know, in the community itself. And so on the left, you see a diagram of where all the privately registered wells are around this facility that could be impacted by this contaminant. People were also giving us resources on what was going on. And so this is one of the workers that was cleaning out an exhaust fan in that room with a pile of treated seed. And he's just covered in neonicotinoid dust, and they just go and air dry him or air blow him. So there's kind of a misconception that because these compounds don't cause cancer and that they are safer than your traditional ones, doesn't mean that it's still safe. And so this is kind of really concerning that the responses of some of the folks that are using these products seem to be, well, it doesn't seem to be, maybe they're not aware of the dangers, or maybe they've been told that these are safer, but certainly that makes me concerned about their health. We put on a number of town halls, created a research team to kind of look into the broader ecological impacts of this, and invited a lot of the senators to come join this conversation. It certainly is kind of strange to be presenting our research with a line of local media, but I think it was really helpful to continually keeping the people engaged. We encouraged our students to talk about the type of research that they're doing and what they were doing. And the community members, I think, were really appreciative of the engagement. And this is Jody, and a lot of the folks were certainly, and rightly so, afraid of what the research team was starting to find. So I bring this up because everyone has a role to play. If you're, the researchers like myself helped identify these areas of concerns, but I reached out to organizations and industries to help establish context. Is this typical? Is this proper? Is this how it's usually done? Come to find out, really, nobody's really talked about large-scale disposal before this. And it wasn't until the journalists wrote about it to bring this issue to the public that the public started to put pressure on the policymakers and the regulators to address this issue. So I attribute everyone to really getting action done in the state. And that was when immediately after the local news coverage, Altent was closed down, and then these bills were enacted to prevent it from happening again. However, even though the plant was shut down, there was a ton of waste on the property, continuous rain and spreading of these contaminants. So we continue to look at these impacts on bees. I had students look at behavioral studies, look at how the behaviors of these bees were changing. And again, similar to studies that we saw in Minnesota, is that with these neonicotinoids, if the foragers are dying away from the hive because of high levels of contaminants in their nectar and the pollen, they are dying away from the hive, causing younger bees to have to precociously forage. So everybody is working harder to compensate for the loss of foragers, to compensate for the lack of workers to keep the colony going. This study is published, so you're welcome to take a look at it. I'm going to be brief because there's so much policy stuff I want to go over, but we saw similar impacts on the queens, on foraging, on food processing. And why this is so important is, in a previous study in my master's days at Washington State University, we looked at the impacts of dirty comb versus clean comb. And this is a waxy substance that can harbor or sequester chemicals within the structure itself. And so this is an example where one of the combs had 17 different compounds. 5 of them were neonics, and you can see the spread of the compounds and their levels. The ones in red are considered highly toxic to bees, yellow, moderately toxic. Now, even though they're classified as highly or moderately toxic, they're not all at levels that would cause harm individually, but the combination and the mixture, well, we just don't know how and which drives the impacts that we see when they are in this really complex mixture. But what we found is that when bees are reared, when they're developing in these dirty versus clean comb, we see things like delayed development where the larvae itself are not developing like they should. And that leads to, again, spotty brood patterning like we saw in those observation hives. But we also see effects of these on the emerging bees. So as they're emerging, some of these bees are becoming later and later in terms of their development. They're coming out as adults a day or day and a half late than normal. The ramifications of that really is tied to mite control issues. And so this probably might make more sense for beekeepers, but the varroa mite goes in and essentially feeds on the host bee during that pupal stage, that cocoon stage. And so this diagram just shows the mother mite going into that capped cell and then starting their reproductive cycle. And that includes a male mite and then the first daughter mite, second daughter mite, and so forth. And so the numbers here basically suggest that the first daughter mite has a 94% chance of survival of coming out with the host bee, but the subsequent mites have lower and lower survival because they have to be fully mature in order to withstand the emergence of that bee. And when you have drone bees, these are the males, they naturally develop 2 to 3 days longer. And so you can see the subsequent daughter mite success for the second mite is 94% as opposed to 38%. So any delay in development actually gives the varroa mite a higher chance of becoming more reproductive and putting out more daughter mites out there. So it does have ramifications for management. The other thing that it has really shown is that when you have these bees come out of their comb, you put them in different challenge experiments, they seem to be more susceptible to other diseases. So nosema or variforma, which is what it's called now, is a microsporidian that affects the midgut of these bees. And we found that if those bees as adults were put into colonies with normal or high levels of nosema, they actually are more susceptible to those diseases as adults. So we individually analyzed every single bee for nosema, and then we started collecting them at different age. So at 2 weeks old, 3 weeks old, and 4 weeks old, after they came out as an adult, we put them in those colonies. In that experiment, in one of those colonies, we were artificially putting in 50 million spores to increase the nosema level, so challenging with a high nosema load. What was really interesting was the bees that are reared in clean comb really showed low levels of infection, whereas the treated dirty comb bees, it didn't matter if they were in a high or a low nosema colony. It was more of how vulnerable those individual bees were, not their environment. So when we look at all of the data put together, when we sampled them at 2 weeks, 3 weeks, and 4 weeks old, turns out that not only are there more number of bees infected, and that's the proportion affected in the bars, the levels also increased over time. So generally thought of as, in this case, nosema generally affects old foragers. At least that's what a lot of literature says. In this experiment, just altering the environment in which the babies were born affected the susceptibility of those adults in which they were becoming infected at a younger age, and those infection levels were increasing over time. And so it goes to show that there is a lot of these subtle, chronic, indirect effects that we really don't have a good understanding of when there's so many things going on in the hive itself. So going back to this Alton project, we were really interested in this idea of dirty comb, right? A lot of beekeepers will reuse comb from a dead colony to restart a new colony. And we wanted to see, well, does this affect the ability of the queen, of the colony to rear a replacement queen and to have a queen come out, mate, and start laying worker eggs again? And yes, it does. It affects the number of queen cells that they produce. And Rogan Tokash was a master student here that did this work. He replicated using dirty comb and clean comb. The bees were the same, the eggs were the same, and so they should have had very similar setup. The only difference was whether or not they were given these dirty food frames from Altent site or dirty or clean food frames from another dead colony. And it turns out that the proportion of clean colonies were able to successfully requeen was 82% compared to 32% of the contaminated colonies. This is a huge difference. And ultimately, he did this in different locations to really kind of suss out whether or not this was coming from the food stores or the locations. And what we found is that the requeening failure was mostly associated with the reusing of the food stores and not the location itself. The other thing I have another student working on now is looking at this idea of varroa populations in these two locations. She is looking at a drone brood trapping mechanism, so utilizing the biology of the drone's development, and mites tend to prefer to go into drone brood because of that longer development time. And what she is finding is that the mite loads are higher in these pesticide contaminated locations. And it might be attributed to their decreased varroa or decreased hygienic performance, like we showed in that previous graph from Minnesota in those observation hives. So this is kind of ongoing research that we're going to continue to dig into. But we certainly think that there's a strong connection between the environment and the pesticide loading in these environments as it relates to the mite control and the ability to control those mites. So in this example, the burden of proof is way too high. I mean, you have to spend a lot of money to get the amount of data. The beekeepers don't have the resources, time, and expenses to do this kind of work. I could barely do this work. But ultimately, the messaging is kind of restricted when we just talk about bee effects. And when we broaden it to looking at through a One Health lens, it really kind of shows that the systemic pesticide pollution issue is much bigger than just bees, right? The pollution issue that we are experiencing is actually caused by regulatory loopholes at the federal level because of the exemption of these treated seeds and having very, when in the life of a treated seed does it become waste if it's exempt, after at least it's the seed company. And so that was a real problem that continues to be problematic in terms of regulatory things. But what was surprising to me was that with this EPA, there's no regulatory division at EPA to address this kind of pollution in plants, which is crazy. There's contamination, there's robust regulations in air, water, and soil, but not in plants. So that's where the local and state solutions really become important. And in this experiment, in this study, we really wanted to try to figure out where these animals were being exposed, whether it's the plant, the air, the soil, or the water. Where that highest exposure will tell us who's at highest risk. So for our insect pollinators, that's going to be plants and air. But EPA doesn't monitor non-target plants, so they're missing 1/4 of the possible exposure routes. Now, in a lot of the ecotox studies, we have a lot of information and weight of evidence that it affects individuals, populations, and communities, and they occur at low doses or after acute exposures. For mammals, it's a little bit harder. They're less sensitive, there's less studies, and the effects usually occur at high doses or prolonged exposures. But there's also a lot of knowledge gaps within the population and community levels of these higher order animals. But what we can say is that these arthropods are bioindicator species and they're critical food web supports for the higher animals. So the loss of food support may actually impact these animals in the long term. And so that requires a lot of long-term sampling. So I've never seen anything like it before, but this is a thistle growing in one of those culvert ditches, and I got distracted by the bees. But upon looking at this plant, you actually see every single order of types of insects dead on the plant, from your very resilient flies to your hypersensitive green lacewings and ants and wasps. And so it was just very strange to see that everything feeding and touching this plant was essentially dead. So this One Health team looked at the soil, water, and air, the birds, the frogs. We've got people looking at the nematodes. I'm looking at bees and plants, and the human health concerns. So if you want more information, there is a website with a lot of the results and updates from our One Health team. But to give kind of a sampling, we found pesticides in the tadpoles. We found pesticides in the birds. The UNMC group collected indoor swipes of cabinets and found even after a year, after the plant closed, they're still finding detectable levels of chemicals in the cabinets, as well as when they suck the air out of the homes. They were also finding detectable levels of pesticides in the air in their homes, which is frightening. Absolutely frightening. The UNMC team also recently published their study on the urine samples collected amongst the residents that submitted samples, and several of them exceeded the normal range, and about 30% of them had at least one neonic in there. Some of them were 2 to 20 times higher than what they were considered normal. And of course, again, these samplings occurred several months, almost a year after the plant closed down. So when we talk about bees, there's a lot of sublethal effects that have been outlined, from the behavior to the foraging to the reproductive issues. But a lot of times the regulatory action doesn't occur until the colonies are dead, or there seems to be a much more obvious impact on the colony. Now, people become a little bit more nervous when we move into other animal systems, like the furry ones that we care about, our pets, our wildlife, and even our children. So if we're moving up the scale in terms of exposure, chemical exposure of neonics can cause changes in weight, susceptibility of diseases, respiratory issues, even problems with reproduction and cognitive function. Unfortunately, these are not cancer inducing, and so they seem to be less emphasis on the concern related to these compounds. For me, though, these are a lot of these quality of life issues: asthma, respiratory issues, Parkinson's, Alzheimer's. It doesn't have to be cancer to cause me concern, because these quality of life issues are just as important to me. And unfortunately, we don't have safe benchmarks for these compounds, especially when they come in a mixture and in multiple routes of exposure. So some of this research tying into other animals have shown that, bats, for example, become predisposed to white nose syndrome, a fungal pathogen, much like bees become predisposed to nosema, which is a microsporidian, very close. So what are these other connections that we should be considering? There are studies that are starting to look at that One Health lens. This study was linking the collapse of bat populations with infant deaths, saying that when bat populations crashed because of this white nose syndrome, the farmers actually increased their insecticide use by 30%. Leading to a 8% rise in infant mortality in these areas, because the bats were no longer controlling the insects that were causing problems in these areas. There's other connections I think we should be considering in terms of avian flu and all the other zoonotic diseases that seem to be outbreaking more and more and becoming more frequent than would be normal. Now, I think the previous speaker showed this graphic, but this is just kind of kind of an estimate of how quickly this class of neonics became widespread within our landscape, especially from the early 2000s into the mid 2000s. Unfortunately, a lot of our documentation on the use of neonics stopped around 2015. So if you look at the graphics in 2011, they estimated 4.7 million pounds were being used. And then that dramatically drops because beginning 2015, they no longer they discontinued monitoring seed treatment applications because of complexity and uncertainty. And this is the same kind of language that we saw officials state that they couldn't conclude that the water or the wet cake applications would not result in unreasonable adverse effects on humans or environment, given that there's no uniformity or limit or number of chemicals present in those seeds. And so on one case, they're no longer monitoring it. On the other case, they can't rule out impacts because of how complex it is. So I feel like this class of chemicals and this particular use is not being regulated in a way that keeps us safe, or even we don't even have the information to know whether or not we're at risk, and when and where. And so as we're looking forward, erratic weather, climate change is going to put more pressure on the landscape. It encourages insects, particularly pest insects, to populate and grow faster, become more resistant to chemicals. They spread, they vector diseases. This isn't just a problem in agricultural areas, but in urban and open. So the response is, when these insects come into our area, become moving into areas they're not supposed to, or increasingly become more problematic to control, people are spraying more, mixing more. They're making stronger tank mixes, which are not regulated for ecological risk, and they're even bringing back harsher compounds to deal with this. And so we're continually going down the wrong road with more and more use and more chemicals. And we can see this in the pollen and the beeswax that is sampled in beehives. Remember, these are like canaries in the coal mine. So there's a lot of chemicals that are being picked up by the bees. Some of it is from the beekeeper applied products to control mites, but the majority of it, the insecticide, herbicides, and fungicides, are coming in from the environment. And we can see that in my state, we have pretty high pesticide prevalence, diversity, and concentration. And so this is of particular concern for me. But across the US, you can see it in the water. I think the last speaker talked a lot about this in the water, so I won't belabor it. But we do see that the water testing shows that imidacloprid, one of these neonics, does exceed the benchmark for invertebrates, both in the chronic level as well as some of these acute levels. On the Altent site, this is Mr. and Mrs. Kaiser's pond. They have a pond that's 6 miles downstream, a catch stream for all of this waste, and they reported a number of fish kills, multiple years of fish kills. And so one of the really fascinating things that I heard in the discussion is that when his pesticide results for his pond came back, and this is a property that I found multiple dead animals on, but this is the pesticide results. The neonics, clothianidin, and dimethoxin were reported there. And they said basically, well, the levels were not high enough to kill fish, so you're good to go. But they neglect to say that, well, or at this time with Mr. Kaiser, showing them the invertebrate acute and chronic levels, it certainly exceeds that. So what it means is that even though it's not directly killing the fish, it's killing everything they need to feed on. And so that's another example of an indirect effect that sometimes gets neglected. For the ABC, they were folks that I reached out to talk to them about the concerns with these birds. Surprisingly, there's a threshold that needs to be reached before federal reporting can require. So with herding animals, it's 50 more mammals. For birds, it's 200. Flocking birds, 50 songbirds or 5 raptors. And then for bees, no reporting requirement, which is fascinating. But just like bees, bird toxicity is dependent on the species and the exposure. There's really no clear pattern of sensitivity because it's context specific, where they're living, what are they using, and what are they feeding on. In their resources, they talk a lot about what the potential risk of these treated seeds are to the birds. And in the dark red, I've got kind of the endpoint range outlined so that it's convertible. It's in parts per billion, so you can kind of refer back to that 50 parts per billion or 100 parts per billion that affects bees. But for birds, you're seeing not a whole lot of seeds required to either kill 50% of the population or start to affect reproductive abilities. So it's not simply affecting the bees, but also the birds that rely on these bees. And so with the bird research, there's been a number of studies that show that the breeding populations of birds are crashing. Even some of the common species have been reduced significantly. In the first report from 2019 that I'm showing here, there was a lot of landscape drivers, but it also included cats and cars. 5 years later, they redid this study and submitted a report on the status of birds, and they're finding, that they're still declining, and that about 50% of this 261 species analyzed showed 25% showed significant declines. And those hot spots where the accelerated abundance declines were occurring were in regions with high intensity agriculture. So cropland, fertilizer use, loss of habitat, and pesticide use were definitely strong contributors to those words. And there's a lot of studies related to birds. And so you can pull together all the bee studies and all the bird studies, and they're very common in terms of some similarities in terms of the rapid decline, some of the effects include, reduced body fat and functionality. I really don't know about the migratory birds, but there can be delays in their reduced migratory survival and success. And so there's a number of these parallels that we see in both the birds and the bees. All of this recognition has led other people to kind of reevaluate the use of these and whether or not they are actually beneficial. And so many of these articles are talking about how it really isn't showing the benefits that it was marketed with, that some of these farmers are just breaking even, that it's not the farmers, but the companies that are making money here or getting the benefit. So there's been a number of petitions, lawsuits, and demand for science policy involvement in these new bills. And what I've learned is that um, treated seeds and the value of treated seeds are often discussed in terms of benefits versus harm. And when you're talking to policymakers or legislators, benefits gets distilled down to profits, right? The profitability of farmers versus the harm to the ecosystem and the environment. And that's an apples to oranges kind of argument that we will always lose at, because you're asking them to think about, outweighing the need for wildlife and the environment over some of their constituents and having them to explain why they voted that way, makes them puts them in a difficult situation. But if we change the conversation to not profits versus harm, but need versus harm, that changes the entire stance. We're not against farmers and we're not against profitability of farm operations. What we want to do is establish need. Is the need for these products outweighing the harm it causes in the environment? And currently, the research is showing no. The benefits that come out of these products does not really affect the farmers. So why would farmers not have a choice in treated or untreated seed, right? We want to give them the opportunity to choose. We are with farmers. We don't want to remove tools, but what we do want to do is get rid of things that don't work, that aren't benefiting them. So if we're talking about profits versus expenses, we need to be considering the expense of the disposal and taking care of the ramifications of these ecological and environmental consequences, and not weighing that with the profitability, right? So this has led to a lot of regulatory changes. A lot of this has been extended producer responsibility bills, Minnesota, New York, Vermont, and more recently, Colorado has been going through these discussions. And there's been a lot of support from not just beekeepers and folks that are worried about pollinators and bees, but pediatricians, organizations. A lot of these folks are understanding that because of those federal exemptions, there really needs to be an emphasis on those local and state solutions. And one of the most creative ways I've seen people message this is that the ABC tested the congressional dining hall food and showed them how much of these neonicotinoids are actually in their foods, which was amazing. I thought this was a really great messaging point that 91% of the foods that they're eating in the cafeteria have presence of neonics, despite the fact that most of the food services are committed to sustainability and emphasizing organic foods. So where is it coming from, right? It's because we're not monitoring or testing a lot of the non-targets and assuming that it isn't moving when it is moving all over the system. Many of them are at levels that would be considered dangerous or cause problems for pollinators. And so that's that red versus the orange. And so if we're eating foods that have levels that could harm pollinators, those at the flowering stage would also have levels that would harm some of those pollinators. And we're seeing that not just in the congressional dining hall, but we're finding that there's a lot of foods out there with chemicals that they shouldn't have. The food companies try to do a concerted effort to reduce the chemical loading in some of the food products, and we're finding that unfortunately it's in our cereal, it's in our fruits, it's in our vegetables. It's in a lot of the different foods that we eat. And the problem is the overuse and overdependency of chemicals. This growing issue of resistance is only causing people to add more and mix more and spray more. This is why we have to get involved with policy and advocacy efforts. Contact your legislators, volunteer your time, stay informed, speak up, write your own opinion pieces. A lot of beekeepers were told that if they want action, then they have to take a legal action in order to really stimulate state and federal change. And that's exactly what's happening. We've got a number of beekeepers, Ontario and Quebec beekeepers, that are putting on an action lawsuit. There have been other litigation settlements towards the EPA as well as state Department of Ag. So the more NRDC and a few other Minnesota groups have sued the Minnesota Department of Ag, and they've just recently settled. And there's been a lot of these appeals and litigations dealing with neonics, particularly, have not been monetary settlements, but really stimulating change in regulatory compliance and project and so where the money is really hitting these chemical companies is in the glyphosate and the Roundup and the dicamba lawsuits. They are losing billions of dollars in litigation due to damages related to that. The legal arguments here is that these companies knew of the dangers but failed to label their products correctly. So this failure to warn argument is primarily the argument that many of these are going towards. The defense on the chemical side, and some of them, including Bayer and the other folks, basically have said that EPA approved their labels. And so the state level lawsuit should be dismissed because they followed EPA approved labels. Now, why this is important is because at the federal level, there's been several attempts to increase or to introduce bills that protect agrochemical companies from legal claims, saying that EPA should be the sole authority of pesticide regulations and states and local government should really not be adding further restrictions. So this effort got shot down in 2023, but a number of other similar state bills have been introduced over the years. And this includes a Kansas one that was introduced on a Tuesday and then voted upon on a Wednesday. And so that gives you just a sense of how quickly some of these things are moving before anyone can even talk about it. And for those who are not aware, the 2026 Farm Bill also has some of these language pieces written in. Pesticide immunity, that would give them the immunity to not, the failure to warn argument would no longer be valid in that sense. And they're also trying to, again, give EPA sole authority, so dismissing or kind of restricting that local state restriction. And then again, there's the legal side of things. I have the Pollinator Stewardship Council, NRDC, and all the other folks down below are more versed in all the different types of bills and actions that are going on. So if you want more information, I would strongly, I can give you contacts for these folks that are really staying up to date on this, because a lot of this stuff is moving so fast, it's hard to keep track. But one thing I do want people to kind of be aware of is through the litigation, through the discovery and bringing out these emails, the 2017 litigation actually showed emails that Monsanto Ghost wrote, a really influential 2000 review paper that was used to I guess, support the marketing of this product in the first place. And what ghostwriting is that they wrote significant portions of the paper without disclosing their role and without disclosing that the other authors were given money to partake in this publication. And so based on these findings, it's very rare, but based on these findings, the editors retracted the paper last December. And this is, again, some of the things that I think is making those lawsuits successful for the plaintiffs against the glyphosate. However, the messaging to the public is very, very, skewed. In the second paragraph here, it says that farmers should be aware that activist-driven falsehoods and litigation industry attack campaigns continue to generate uncertainty about essential crop production tools like glyphosate. And so this is an issue. Like the messaging part of it is telling farmers that these actions are making it uncertain that they'll have the tools that they need to keep their crop production going. But it's not just a pesticide exposure problem that we face, it's an agriculture problem as well, because the predominant crops that are being grown in the US um, are not necessarily pollinator dependent, right? And in our state, we have about 49 million acres, but I think 90% of it goes into agriculture. We've got a lot of the Great Plains prairie region that is being lost due to the conversion of grasslands into functional urban and agriculture. Um, and there's a lot of value, economic value for these kind of commodity crops. But there's room for cover crops and other types of pollinator-friendly crops. We're just not giving farmers the resources to incentivize those. So again, in our state, 91% is dedicated to either pasture or corn and soy, leaving very little for any other kind of forage. And so a lot of these efforts to encourage floral provisioning through cover crops, prairie strips, hedgerows, we push it, but adoption is quite low. Even though we're putting millions of dollars into investing on implementation, adoption is quite low, and that's because there's a lot of financial risk, complexity, and lack of knowledge related to this. But I think a big driving force is that the cover crops itself, estimated equip pay, doesn't really cover a whole lot of the environmental quality incentive program, the EQIP programs. You can enroll your land in through the USDA and RCS contracts, and it can cover 60 to 80% of the establishing of the CRP lands, for example. These conservation lands often are really, really beneficial for wildlife and last year, we had about 548,000 acres under EQIP contracts. These are those conservation programs, right? That kind of helps create forage and habitat for various. It just puts pieces of land out of production. But a lot of people don't use these EQIP programs, don't put in these cover crops or put their land into CRP. A lot, I think, is because it's driven by these corn subsidies. They pay way more way more per acre than any of these cover crops or these conservation programs. And if you go back to before these became really, really in 2007, the estimated payback for an EQIP program was between $30 and $70 an acre, and there was 5.9 million acres under contract in Nebraska. So just in the last 20 years, think about the shift from 5.9 million acres to now under 500,000. So this is a huge dramatic change. It's not just the pesticides, but it's also this huge shift in how we use these lands. And I think it's honestly driven a lot by these crop insurance subsidies. So I bring this up because in a lot of these bills and these arguments, the reason why these bills fail is because they don't want to hurt the farmers, that the commodity farmers that depend on these products. But look at the amount of subsidies that they get for crop insurance. And this provides income stability and yield protection and encourages plantings on a larger scale. It also provides a safety net if they want to experiment on new crops, equipment, or techniques, right? So the arguments against these new prescription models that seek to verify need before treating seems to neglect the fact that they have existing safety nets where beekeepers and specialty crop producers do not have access to. They've got small portions of funding, either through disaster assistance programs or through the uninsured program, crop insurance programs. So it's very different. So I know I'm over, way over. Sorry about that. So I'm just going to finish up here. For beekeepers, if this messaging is for beekeepers, yes, Varroa is a problem, but it's not just Varroa and it's not just bad beekeeping. Poor Varroa management doesn't explain the widespread loss of birds, bats, and other pollinators in our environment. You can do everything right as a beekeeper and still not improve. You could buy hygienic designer queens that are marketed as high performing in terms of their mite control. But if you put them in the wrong landscape, they're not going to perform to their peak optimal potential. So the policy changes is what I think is really necessary to drive the practices and encourage adoption of these new ways. And the bee losses themselves, it's not the problem. They're just the messengers, a harbinger of broader ecological stress that ultimately will affect our agriculture and our food systems and our own daily lives. So, to sum it up, food security is not just about quantity, but it has to be about quality as well. All right. So my take home is that we need to come together. We need to stop fighting amongst ourselves, the hobbyists versus commercial, the wild versus managed bee pollinators, the farmers versus gardeners versus conservationists. There's a lot more that we have in common than we think, and that is focusing on forage and habitat and reducing chemical exposure. So I know I went through a whole lot. I was really trying to get as much information as I could. So I hope that people kind of learned a lot, get fired up, and try to get more involved. Yeah, that's, that's it. I'm sorry I went way over there. Let's see if I can get my keys back. Ryan, are you? Oh, I don't know
We're
how to do that.
here. Yes.
You guys know how to, oh,
Nope.
there it is.
Don't be sorry. That was awesome. This is like a great repository for this information. Thank you, Dr. Wu Smart. This is wonderful. We do have a few questions if you have the energy
I
to
do,
get through
and
them.
yes, I do, and I was trying to figure out a good way to get the Q&A box and the presentation on the same page, and I just couldn't.
That's
So,
okay.
yeah,
I got a few of them here ready to go
sure.
for you. One that came in was so the Mead, the ethanol plant in Mead has been closed, you said,
Yes,
correct?
correct.
They were wondering, is there other Mead style operations elsewhere in the US?
That's a great question. When we were looking into this project, there was initially a small plant in Leota, Kansas before the Nebraska one, but the EPA came in and shut that down. And then I think they jumped over to the Nebraska facility. And currently, we really don't know where large-scale disposal is occurring. And that I think is a big problem. And the Alton plant shut down in 2021. But ag hasn't changed. The practices haven't changed. So this need for disposal, I think, is still there. We just don't know what's happening.
Hmm.
Yeah.
Oh. Um, s- Scary to think about. Jeepers. We had a question come in early from a rancher in South Dakota. They were curious, and this is a difficult one to answer, I know, but they're curious on how big of a pesticide-free island might someone need to protect their beneficial insects or just their own health for themselves. How would you answer something like that?
Well, honestly, the when we were doing the comb study and looking at dirty comb versus clean comb, it was extremely hard to find any comb that did not have chemicals in it. We found one example from a feral colony in a desert. And so
Hmm.
the reality is that we are exposed to chemicals almost everywhere possible, from open spaces to lands to urban to ag. And so it isn't about creating a chemical-free zone, but reducing the risk of exposure and then mitigating exposure impacts with other things like planting more flowers, providing resources that are clean for the bees, changing out comb if you need to. And so I think it's better to think about the fact that there's no getting away from it. So how can we do what we can to reduce the damages and the effects of these chemicals, right? These are chemicals that are in the air, the food, the plants, the soil, the water, and they come and go with rain, with wind, with these different environmental seasons and factors. So it's almost impossible to predict that exposure risk, but we can be aware of it and mitigate what we can.
Mm, yeah. Yeah. Um, so on that note, we had somebody ask, uh, do these, excuse me, once the neonics are in the soil, how quickly do they move through the soil profile? Is it known which soil bacteria have the ability to break these neonic compounds down?
Yeah, that's a great question. There is some resources out there that look at persistence in soil, and they're finding that it's highly dependent on that soil health, that soil component itself, right? If there's a lot of organic matter, these residues tend to stay in and persist more. If it seems to be loomy with more sandy-like materials, then it seems to percolate and travel and move down faster, so it doesn't persist in the soil, but it has a higher chance of leaching into the groundwater. And so it really is kind of dependent on what is in the soil itself and how much moisture is in that system that can kind of move the chemicals down. In our soil sampling, it was not in the first 8 inches of that soil column. It was lower down because these chemicals also photodegrade. So at the top, they're getting degraded by the sunlight, right? But they're also quite mobile with water. So they're getting pushed down every time it gets rained or there's movement. So that's why at the top layer, there was very low levels of neonics. You had to get down lower to really find them in terms of those that, as high as they express. The fungicides, however, did not degrade, at the top level. You still saw strong levels of fungicide in just, a few inches of that soil column.
Oh, okay. Um, we had a question here about monitoring programs. Are there any monitoring programs ongoing to track biodiversity and abundance of arthropods or other animals in the area over the years after the ethanol plant closed in Mead?
Oh, in our area specifically, I mean, we've got the One Health team, right? So,
Mhm.
unfortunately, the first round of funding came from legislative funds that helped do a lot of the sampling. And now a lot of that funding is gone. So, I don't know if these labs are kind of wrapping up their research. On our end, we don't have funds to continue to put into bees in this landscape and then watching them die. But um, there are some folks that are trying to continue long-term monitoring, particularly the birds. If it's coming in the bird eggs, think about how much of it is actually the birds are being exposed to for those compounds to travel through their tissues and through the chorion of the egg and into the yolk, right? To get those levels, 300 parts per billion in the yolk. And what does that mean in terms of the bird population? So those are animals that probably are going to require long-term support. The humans that live in the community, with a lot of them showing levels in their urine that exceed normal levels, what does that mean in terms of their own health, right? And so the cattle that were right adjacent to that property, unfortunately, those cattle are going for human consumption and they're not tested. We have no access to those facilities. We don't have testing done for those cattle. But more frightening, one of the comments that we were given was that the solution to pollution is dilution. So, meaning that if they are polluted, but they go into the food chain, they get diluted by everything that isn't polluted, and eventually the problem is solved. For me, that is assuming that there's a lot of uncontaminated foods there that will filter and dilute those contaminants. And I don't know if we know that. It on the other side of that property is an organically certified field, right? But a lot of the farmers are afraid to have testing done because they don't want to lose those organic certifications. So I think that the problem is that the overuse and dependency of compounds is rapidly outpacing our ability to understand what they're doing in the environment. And we really have to get control of that so we can prepare ourselves or at least slow down the detrimental effects and consequences of this use. Yeah.
Wow. It's quite the hairball to untangle.
It is. But I
Yeah.
think there's a positive note that, people are starting to become more aware and more engaged, right? And that's what we really need is people to get more involved, more vocal, really think about what we need to do to change it and prevent things like, the changes that might protect an agrochemical company from being accountable and then making sure that the state and the local officials are aware of these things so that they can step in and provide those extended producer responsibility bills or increase the restrictions or provide extra monitoring and enforcement so that we don't continue to have these issues pop up in different states, every few
Yeah.
years. Yeah.
Yeah. It makes me wonder. So is there, maybe for our beekeeper friends that are tuning in, is there something you talked about organization and groups coming together? Is there something out there to help beekeeper groups come together to maybe raise awareness of this issue with their policymakers and lawmakers?
Well, I recommend for beekeepers, because there's so many different beekeepers and all across the U.S. Start at your local and state level. There's local bee clubs, right? That some of them are highly engaged. They select a policy or advocacy representative that helps them keep informed about different things that might relate to bees. Some create committees to enact and encourage pollinator-friendly policies, whether that's, changing how honey is sold or making, some of the choose local produce programs and that help beekeepers market their honey locally and increase their prices. Some people are looking for changes in ordinances that allow them to plant more bee-friendly habitat. And some people are actually advocating for policy changes related to chemical exposure. Some are interested in creating incentive programs to encourage cover crop plantings. So there's a lot of different things that beekeepers can kind of reach out to policymakers and legislators to help them and from habitat to chemicals to opportunities, right, in their own products. So I would say start with the local and state groups, and then there are a number of national groups that they can get involved with. There are multiple national groups, and Pollinator Stewardship Council is made up of a bunch of beekeepers that are really passionate about this issue. So if they have time, they have energy, and they want to volunteer, reach out, say, "Hey, I'm moved by this. What can I do to help?" Right? Because there's only small groups of core people that are in these areas working on these issues. If we can get more dedicated and motivated people to help, the more the better.
Yeah. Yeah, well said. Um, Let me see here. I need to make sure that I've gotten everybody's question. We probably should let Dr. Wu Smart go here soon. Um, let's see. So we did have somebody had a specific question on their bees. They asked, could we use the gestation of brood time to detect dirty comb in our hives?
Not necessarily the gestation of brood time, but certainly if you see a lot of spotty brood or brood diseases in your frames, I recommend beekeepers start marking those colonies or those frames to give them a sense of when they need to trade out their comb. And that's the hardest thing to do. You don't want to replace a bunch of comb without because it's at a cost to the bees, right? It costs time, it costs money, it costs energy, it costs honey for them to replace that comb. So you don't want to do it too frequently, but you do want to have some indicator that, hey, my comb is saturated, it's not performing like it should. Maybe it's time to ditch it. So when we're doing inspections and we see things that are off, you don't want to just throw away all those babies that are developing, but you could mark those frames. And I do the 3 strikes, you're out. So if I have 3 strikes or markings on the top of that frame or that colony, that indicates that I've gone through 3 different inspections and it's still not doing what I think is a standard good, production in terms of brood pattern or these in-tune pollen or other brood diseases. And then when I'm rehiving my packages or when I'm getting dead outs out of my yard, if there's a lot of those markings, it's a good indication that I should just pitch that comb completely. I gave it 3 chances and it just kept failing. And I think that's the best way to encourage beekeepers to start implementing regular combing placement in their operations without just having some bucket number say, I'm going to replace 5 a year every year, because that's a lot of waste if it's not needed. And so let the bees tell you when something is going wrong.
Yeah, thank you for that.
Yeah.
Um, I guess we should maybe wrap it up here. I think all of us are a little shell shocked at all the information you gave us. I certainly will need to rewatch this because there was a lot of good info in there. Um, and to our audience, this webinar was recorded and will be posted to Ecdysis's YouTube channel.
Mhm.
So if you know somebody that couldn't catch it because uh, we're broadcasting during work hours, please advertise because I think this kind of information really needs to get out there to everybody. So please share this with your friends and anybody you think might be interested. Um, and then don't forget, we have a part 3 of this series will be next month in April. I don't have the exact date, but you can look for it on Ecdysis's website. We have a calendar of events on our website, ecdysis.bio. And you can go ahead and sign up for that webinar. It'll be from Dr. John Lundgren, our kind of the head honcho of Ecdysis, and he's going to be reviewing his work on neonics and some of the higher level organisms and speaking to what that what that really means for the larger ecosystem as a whole. So
Yeah,
you'll
he's done
join
some
us
great
next month.
work with some of the larger animals that I was hoping that at some point would be discussed. And I think
Yeah.
it also showcases how much of these compounds spread in a way that we had no idea. And I guess the take home for folks that are maybe thinking it from a farm perspective or farmer's perspective is that the mobility and the persistence of these compounds, right? With the organic soil, they can persist for years. So the persistence and movement of these compounds, what do you think it's doing to chemical resistance issues in farms and when you're talking about mosquito or invasive species control, when you're talking about your own garden control? I think these persistent compounds are exacerbating the issue with chemical resistance developing in pests and pathogens all across our landscape. So we have to really consider not just these compounds, but how they function in relation to all the other things that we're putting into our environment.
Yes, yes, well said.
Yeah.
Um,
Well, thank you.
Wonderful. Thank you again, Dr. Smart. Yeah, this will conclude our webinar for today. I wish we could all applaud, but I'm the only one that's in person. So thank you again for everything you shared with us. That was wonderful.
Thank you.
Yeah.